Welcome back to the deep dive. Today, we are tackling a really foundational chapter from Gray's Anatomy, the world of blood, lymphoid tissues, and haemopoiesis.
It's a huge topic.
It is, and it can be incredibly dense. So, our mission today is to, cut through that and give you a really clear, visualizable way to understand how all these systems are built.
Sounds good.
So let's start with the medium itself, with blood. I mean, it's this opaque, viscous fluid that's just constantly on the move.
And you can tell so much just from the color, can't you?
You really can. It's that bright, almost scarlet red when it's full of oxygen in the arteries.
And then it shifts to that much darker, sort of purplish red in the veins once it's dropped off its cargo.
Essentially, you've got two main parts: the liquid plasma and all the cells floating within it.
Right, and that plasma, it's this clear yellowish fluid, and it's basically the body's entire logistics network.
So it's more than just water.
Oh, much more. I mean, yes, it's mostly water with small stuff like glucose and sodium, but the real power comes from the plasma proteins. These are the active machinery. You have things like prothrombin, which is a clotting factor. You have immunoglobulins for defense.
Like antibodies?
Exactly. And you have transport globulins that are like little Ubers, just ferrying hormones and iron all over the body.
Which is why running blood work is so incredibly diagnostic.
Absolutely.
Yep.
You're reading the body's story in that fluid because every cell releases things into it. We're even moving into metabolomics now.
So that's looking at all the tiny molecules.
High-throughput analysis to spot pathologies way before symptoms even appear. It's the future of diagnostics.
That makes perfect sense. The plasma is the communication highway. Okay, let's unpack the elements actually using that highway, starting with the red team.
The erythrocytes.
The erythrocytes, they make up a staggering 99% of all blood cells.
It's an incredible number.
And to picture one, think of like a donut, but without the hole punched all the way through. It's a biconcave disc.
That shape gives it a paler appearance in the center, which is a key identifier.
Right. And under a microscope, they do this funny thing where they stack up like coins.
Rouleau formation.
Rouleau, yes.
And that biconcave shape, it's not just a random design. The body invests huge energy in it.
It's for more than just surface area then.
Way more.
Yeah.
It's about the internal membrane skeleton. There's this protein called spectrin that forms a lattice inside.
A support structure.
A support structure that gives it incredible flexibility. It lets the erythrocyte squeeze through capillaries that are half its size, sometimes down to just 4 micrometers wide.
And then it just snaps back into shape.
Instantly snaps back. If that lattice fails, the cell is fragile and gets destroyed.
Wow. But the whole point of this amazing cell comes down to the molecule it's packed with, hemoglobin.
HB.
The oxygen carrier. It's a tetramer, so 4 subunits, and each one holds an iron-containing heme group.
And we have different types of chains that make up those subunits: alpha, beta, and in a fetus, you see gamma chains, which gives you fetal hemoglobin, HbF.
And that's where the clinical correlations really come into play, isn't it, when those chains are faulty?
Oh, absolutely. If you have a defect in the chains, the whole system can just collapse. In thalassemia, for example, you might not produce enough of one chain type.
And the hemoglobin becomes unstable.
Unstable, misfolded.
Right.
But even more dramatic is sickle-cell disease.
Right.
The defect there is minuscule. It's a single-point mutation that swaps out one amino acid for another in the beta chain.
Just one. And the consequence is that huge.
It's massive because when oxygen levels drop, the faulty hemoglobin molecules don't stay separate. They polymerize. They stack into these rigid rods.
And that deforms the entire cell. That's the sickling.
That's the sickling. And it causes blockages, pain, organ damage. It's profound.
And these cells, they only last about 100 to 120 days. What happens then?
They get recycled with just incredible efficiency. Macrophages, mostly in the spleen and liver, identify the old fragile ones and just eat them.
Phagocytose them.
Yep. And nothing is wasted. The globulin protein is broken down into amino acids to be reused.
And the iron.
The iron is grabbed by a protein called transferrin and sent right back to the bone marrow to make new cells. The rest of the heme group gets turned into bilirubin for excretion. It's a perfect closed-loop system.
Okay, so since these cells are moving all over the body and used in transfusions, let's talk about their ID tags. The antigens.
The blood groups.
Right, the blood groups. We're mainly focused on ABO and rhesus, clinically speaking.
We are. So the ABO system is all about which antigens A or B you have on your red cell surface.
And the key thing is the antibodies in your plasma.
Exactly. If you're group AB, you have both antigens, so you don't have any anti-A or anti-B antibodies.
Which makes you the universal recipient.
That's the one. And group O is the opposite. No A or B antigens on the cells, so they're safe to give to almost anyone. The universal donor.
Even though their plasma has antibodies.
Right, but those antibodies get diluted out during transfusion, so it's generally safe.
Okay. But the rhesus system is where it can get really dangerous.
It is, especially with the D antigen. Here's the critical detail you have to remember. Rhesus antibodies are a specific class, IgG.
And IgG can cross the placenta.
It can cross the placenta. So if you have an Rh-negative mother carrying an Rh-positive baby, she can become sensitized. Her IgG antibodies can cross into the fetal circulation,
And attack the baby's red blood cells.
causing massive destruction. Hemolytic disease of the newborn. It all comes down to the fact that IgG is small enough to cross that barrier.
That's a huge why you should care moment for understanding antibody classes. Okay, let's shift from transport to defense. The leukocytes,
White blood cells.
the immune troops. And we start with the granulocytes, and the big one here is the neutrophil. They can be up to 75% of your white cells.
Sometimes called polymorphs.
And the visual key is that nucleus, right? It looks like it's in 3 or 4 separate lobes.
Segmented, yeah. And that weird shape gives it the flexibility to squeeze out of blood vessels and get to the site of an infection.
So once it gets there, what does it do?
It has two main weapons. First, it's a voracious phagocyte. It just engulfs microbes and kills them with a burst of what we call reactive oxygen species.
So chemical warfare.
Basically, yeah. But the second method is even more dramatic. It deploys something called a Neutrophil Extracellular Trap, a net.
What on earth is that?
So think of it like a spider web. The neutrophil literally expels its own DNA and enzymes in a final suicidal act
To trap bacteria.
to trap and kill bacteria. It's an amazing defense mechanism, but it costs the cell its life. They only live for about 6 or 7 hours in the blood.
And that's what pus is, isn't it? Dead neutrophils.
Dead neutrophils, cellular debris.
Yeah.
And that greenish-yellow color actually comes from one of their enzymes, myeloperoxidase.
Incredible. Okay, next up in the granulocytes, the eosinophils.
Smaller numbers here.
And visually, they're really striking. They have a bilobed nucleus and these large granules that stain bright pink or red.
Unmistakable. Their main job is tackling targets that are too big to eat, like parasitic worms.
So they don't phagocytose them.
No, they release toxic proteins, like major basic protein, directly onto the parasite surface to kill it. They also help limit inflammation.
And the last and rarest is the basophil, less than 1%.
Right. They're defined by these huge dark granules packed with histamine and heparin.
Inflammatory agents.
Yep. They're basically the circulating version of tissue mast cells. They have IgE receptors, and when an allergen binds, they release everything and trigger those immediate allergic reactions, like hay fever.
Got it. Now we move to the mononuclear cells. First, the monocytes.
The biggest of the leukocytes.
And they have that large, sort of kidney bean-shaped nucleus.
They do. Monocytes are the ultimate transformers. They are phagocytic in the blood for sure, but really they're just passing through.
On their way to the tissues.
Exactly. Once they leave the bloodstream, they differentiate into the real long-term specialists, macrophages and critically antigen-presenting cells, or APCs.
And those APCs are what kick off the next phase of the immune response.
The adaptive response, yeah.
Which is run by the lymphocytes. The second most numerous type of white cell. They're small, and the nucleus takes up almost the entire cell.
A very high nuclear to cytoplasmic ratio when they're resting.
But their unique feature is that they recirculate, right?
Yes, this is crucial. They leave the blood, they go into tissues like lymph nodes, and then they return to the blood. It's a constant surveillance patrol.
This is where we get into B cells and T cells.
The adaptive arms. B cells are our antibody factories. When they get activated, they transform into plasma cells.
And those look really different under a microscope.
Very different. They're packed with rough ER to make all that protein, and it gives their nucleus this classic clock-faced appearance. The chromatin is clumped around the edge.
So those antibodies come in different classes?
They do. IgG is the main one in circulation, and it's the only one that crosses the placenta.
Providing that passive immunity to a baby.
Right. IgM is the first responder in an infection. IgA is the secretory one. You find it in saliva and tears, protecting mucosal surfaces.
And then we have the T cells, cell-mediated immunity.
And we define them by their CD markers. CD4 plus cells are the helper T cells. They're the coordinators, the generals. They secrete cytokines to direct the immune response.
And the CD8 plus cells.
Those are the assassins. the cytotoxic T lymphocytes, or CTLs. They hunt down and kill infected cells like virus-infected cells.
How do they do it?
They release a protein called perforin, which punches holes in the target cell's membrane.
A perforation.
Exactly. And then they inject granzymes through that hole, which tells the cell to undergo apoptosis to commit suicide.
And you've also got regulatory T cells to keep everything in check.
The brakes on the system to prevent autoimmunity, absolutely critical.
It seems like there's a cell that bridges the gap between that innate immediate response and the adaptive T cell response.
You're thinking of the natural killer cells, the NK cells.
Right.
They're functionally a lot like those CTLs. They use perforin and granzymes too, but they don't recognize a specific antigen. They operate on a missing self mechanism.
What does that mean?
It means they kill any cell that lacks the self-ID molecule, MHC class I, on its surface. Viruses and cancer cells often try to hide from T cells by getting rid of their MHC.
But in doing so, they make themselves a target for NK cells.
It's a brilliant complementary system, an amazing backup.
Okay, we've covered the fluid and the defenders. We need the repair crew. Platelets.
Or thrombocytes.
And these aren't even real cells, are they? They're fragments.
Tiny anucleate fragments shed from these absolutely giant cells in the bone marrow called megakaryocytes.
And their entire job is haemostasis, stopping bleeding.
That's it. When a vessel gets damaged, they stick to the site, they aggregate, and they kick off the clotting cascade that ends with a stable fibrin clot.
And then they actually contract.
They do. Their internal cytoskeleton contracts, pulling the edges of the wound together. We call it clot retraction.
So we've seen the cells. Let's look at their headquarters, the lymphoid tissues.
We split them into primary and secondary.
Primary is where they're made, so bone marrow and the thymus.
Right. And secondary is where the immune responses actually happen. So lymph nodes, the spleen.
Can you walk us through a lymph node? Help us visualize it.
Okay, so picture a small kidney bean-shaped encapsulated organ. Lymph fluid comes in through multiple vessels on the outside edge, the afferent lymphatic.
Into a space just under the capsule?
The subcapsular sinus, yes. Then it filters down through the node and finally collects and leaves through a single vessel at the indentation, the hilum.
The efferent vessel.
The efferent vessel. And it's organized into zones. The outer part, the cortex, is B-cell territory. That's where you find the follicles.
And deeper in.
The pericortex is T-cell rich. And crucially, it contains the high endothelial venules, or HEVs. These are the special doorways for lymphocytes to enter the node from the blood.
So that's the main entry point for the cells?
For circulating cells, yes. Then the center, the medulla, is full of plasma cells just pumping out antibodies into that exiting lymph.
What about the defenses that aren't in these neat little packages?
That would be MALT. Mucosa-Associated Lymphoid Tissue. It lines our gut, our respiratory tract.
Right at the front lines.
Exactly. And the key structural difference is that MALT has no afferent lymphatics. Cells can only get in from the blood through those HEVs. Its whole job is to secrete IgA to protect those mucosal surfaces.
Which brings us finally to the factory floor. Haemopoiesis, the creation of all these cells.
Postnatally, this all happens in the bone marrow.
And in adults, it's not in all our bones, is it?
No. Active red marrow is restricted to the central skeleton. Your sternum, ribs, pelvis, and the very tops of your femurs and humeri. The rest of the long bones fill with fat yellow marrow.
And it all starts from a single pluripotent stem cell.
A very rare stem cell that then commits to either the lymphoid or the myeloid lineage.
Let's trace the red cell pathway, the erythroid line. That's controlled by erythropoietin from the kidneys, right?
Tightly controlled by it. The process goes through a few stages, but it ends with this one incredible step: nuclear extrusion.
The cell literally pushes its own nucleus out.
Actively ejects its nucleus to become an anucleate reticulocyte, which then matures in the bloodstream.
And the platelet lineage is just as wild.
It really is. The megakaryocyte, that giant parent cell, doesn't divide. It just replicates its DNA over and over, becoming massively polyploid.
And then?
It extends these long arms into the bloodstream and just sheds thousands of little cytoplasmic fragments, which are the platelet.
Incredible.
And we have to finish with maybe the most stringent quality control system anywhere in the body: T-cell selection in the thymus.
This is where they get educated.
They get educated, and it is a brutal process. They have to learn to recognize self-MHC molecules, but they absolutely must not react to self-antigens.
So they don't cause autoimmune disease.
Exactly. The process is so tight that something like 95% of all T-cell progenitors are killed off by apoptosis before they ever even leave the thymus.
What a journey. From the flexibility of a single red blood cell through the chemical arsenal of the neutrophils, the adaptive power of lymphocytes, and finally to the factory floor itself.
The big takeaway for me is just how integrated it all is. You see how the fluid dynamics, the cell architecture, the genetics, it's all one interconnected network.
And a tiny flaw can just derail the whole thing.
A single point mutation in hemoglobin, a mistake in T-cell selection, and you have a profound systemic disease. It's a system that runs on precision.
Thank you so much for joining us on this deep dive into the anatomical basis of blood and immunity.
And as you go about your day, just consider the elegance of that regulation. What is the invisible command structure that allows the bone marrow to produce 500 billion red cells every single day, perfectly matching the number destroyed?
A perfect balance.
Think about those simple feedback loops, those signals like erythropoietin that maintain that critical, life-sustaining stability on such a colossal scale. It's just amazing.
Absolutely fascinating. We hope this deep dive helps you master these foundational concepts. We'll catch you on the next one.